Hi everyone. Today we will discuss DNA replication, transcription, and translation. Let's go over the central dogma again. DNA replication is a process that cells make another copy of its genomic DNA, so it is always associated with cell division, including mitosis and meiosis. Using DNA as a template to make RNA is called transcription, and making protein from RNA is called translation. Remember that DNA replication and transcription only happen in nucleus, and translation only happens in cytoplasm. Now let's discuss DNA replication in details. We learned that DNA replication occurs in S phase of cell cycle. DNA replication is a semi-conservative replication, which means two copies of the original DNA molecule are produced. Each copy contains one original strand and one newly synthesized strand. It is important to remember that DNA replication proceeds in a 5' to 3' direction, and it occurs only in S phase. Let's watch a video on DNA replication.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
In the video, there are several enzymes that are essential for DNA replication. These include DNA helicase, which unwind the DNA double helix, RNA primase, which builds an RNA primer on the parent strand to initiate DNA replication, DNA polymerase that matches and lays down nucleotides to build the daughter DNA strand along each parent DNA strand, and DNA ligase that joins the adjacent Okazaki fragments on the lagging strand of DNA. It would be nice to remember all these enzymes and their functions because they will be tested in the exam, all right? Again, I would like to go over a few slides to repeat several key points. First, during DNA replication, two polynucleotide strands uncoil, and each is a template for synthesizing a new strand. A replicated DNA molecule contains one new strand and one old strand. This mechanism is called a semi-conservative replication. Another important feature is the direction of DNA replication. We learned that each DNA strand has two ends, 5' and 3'. So DNA polymerase can add nucleotides only at the 3'. Therefore, DNA synthesis proceeds only in the 5' to 3' direction. Because DNA synthesis can proceed only in the 5' to 3' direction, only one DNA strand can be assembled in a single piece. For the other strand, newly synthesized DNA is made in short segments and linked together by the enzyme DNA ligase. These short segments are called Okazaki fragments, okay? So now let's watch another video to learn the process of transcription and translation.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
This slide shows the flow of genetic information. One strand of DNA is transcribed into a strand of mRNA. The mRNA is processed and moves from nucleus to the cytoplasm, where it is converted into the amino acid sequence of a polypeptide that folds to form a protein. Now let's learn some detailed information of transcription. Transcription is such an important process, and then we will keep discussing it in the next 2 lectures. Transcription has 3 major steps, namely initiation, elongation, and termination. Transcription begins with the binding of RNA polymerase together with one or more general transcription factors to a specific DNA sequence on the promoter region. Okay. Upon binding, RNA polymerase helps to unwind the DNA helix and prepares for transcriptional elongation. During transcriptional elongation, RNA polymerase further unwinds the DNA helix, and RNA polymerase reads the template strand and synthesizes the pre-mRNA molecule. Transcriptional termination is a process that release RNA polymerase and pre-mRNA from the template DNA. Once the transcription is completed, DNA strands reform into the double helix. This slide shows the 3 major steps of transcription. The RNAP showing here is the RNA polymerase. It actually contains a protein complex, many proteins in this complex. Notice that transcription factors and their associated transcriptional co-activators bind to the region upstream of transcription starting site, this TSS. So this region actually include both promoter and enhancer. Okay, in this picture, this point has been simplified. Also, please note that both transcription and the translation follows the direction from 5' to 3', similar to the DNA replication. Transcription produces pre-mRNA, and the pre-mRNA, you can see the 5' cap and the 3' poly A tail. These are added to the ends of the pre-mRNA molecule. Meanwhile, this splicing occurs to remove the introns in the mRNA, the pre-mRNA, and producing the mature mRNA. Okay, so the mature mRNA is then moved to the cytoplasm for translation. This is splicing. In pre-mRNA processing, introns are spliced out and exons are combined in the mature mRNA. However, the splicing may not always produce the same mature mRNA. Sometimes, particularly exon of a gene may be included within or excluded from the final mRNA produced from that gene. So consequently, the proteins translated from alternatively spliced mRNAs will contain differences in their amino acid sequences. We call this process as alternative splicing. So alternative splicing allows for one gene to code for more than one protein and explains why more than 20,000 proteins can be produced from 20,000 genes in our genome. Actually, the proteins is far more than 20,000. We'll give you the number next. So this slide shows what alternative splicing really means. In the pre-mRNA processing, introns, okay, they are spliced out, and exons are combined together. Okay, those are included, all the extron here, showing this protein, they make protein A. But in alternative splicing, some exons are removed during processing, resulting in the production of different proteins. For example, in protein B, the exon 4 is not included. So exon 4 is removed during the splicing. And in protein C, the exon 3 here is missing. It's also removed during splicing. So different splicing, they produce different proteins. Okay, now let's see how protein is finally made by translation process. We all know ribosome is where translation occurs. Ribosome consists of two major components. One is the small, the ribosomal subunits, and a large ribosomal subunit. Okay, so in eukaryotes, scientists have identified 60S as the large subunit and 40X as the small subunits. Each subunit consists of one or more ribosomal RNA, that's also called rRNA molecules, and many different ribosomal proteins. Okay, so this slide shows a three-dimensional models of the small and large subunits of ribosome. The tunnel showing here, is where the growing peptide chain exists, the ribosome during translation. All right. But then one special feature of ribosome is that ribosomes are they're also called ribozymes, okay, meaning that RNA molecules can have the ability to catalyze the specific biochemical reactions. In general, we consider enzymes are always proteins, right? But the large subunit ribosomal RNA can catalyze and link amino acids together during the protein synthesis. So RNA can also function as enzyme. So please remember this exception, all right? The transfer RNA or tRNA is another important molecule for translation. So each tRNA contains a region called this anticodon region that recognizes and binds to its complementary codon in mRNA. At the other end here is a region that binds to amino acid specified by the anticodon or codon combination, all right? So as we learned that RNA can also form certain structures, right? So the tRNA is an RNA molecule, right? So the secondary structure of tRNA will look like a clover leaf. So you see this hydrogen bonds between these complementary nucleotides stabilize the RNA structure. Here, I would like also to introduce the concept of wobble bases. Wobble base pair is a pairing between two nucleotides in RNA molecules that does not follow Watson-Crick base pair rules. Okay, here shows the four main wobble bases pairs, G to U or U to G, same. And then I to C or I to A or I to U, okay? So in tRNA, this I stands for inosine. This is a nucleoside, which can pair with CAU on mRNA. It is interesting to note that only the first two bases of the codon have a precise pairing with the basis of the anticodon of tRNA, while the pairing between the third basis of codon and the anticodon may wobble. So this allows a single tRNA to recognize more than one codon. Okay, therefore, although there are 61 codons for amino acids, the number of tRNA is far less. Actually, it's around like 40. This is due to the wobbling. So showing here is an example, all right? So the tRNA has an anticodon of a GAG, which is, then this codon forms a normal pairing with the mRNA, recognize this is CUC sequence, and brings in this amino acid called the leucine residue, okay, leucine. However, this is the same tRNA can also recognize a sequence of CUU on the mRNA with the third codon is wobble pairing, G paired with a U, and brings in the same leucine residue, all right? So because anticodons on tRNA recognize the three nucleotides on mRNA each time, the genetic code are triplet codons, okay? The codon to start the translation is a start codon, which is always AUG, I show right here, encoding the amino acid called the methionine. Interestingly, the three-letter codons direct the assembly of exactly the same amino acids in nearly every organism on Earth, like bacteria, plants, and human beings all use exactly the same genetic code. We say that the genetic code is universal. Meanwhile, the genetic code is redundant because the same amino acid residue can be encoded by multiple codons. This is due to what? This is because the wobble pairing we just talked about. So this table shows the genetic code. I would like you to remember that this methionine here is always the first amino acid to make during the translation. And the stop codons has three of stop codons showing here, UAA, UAG, and UGA, okay? Here we have 20 different amino acids in total that can make proteins. Then how these amino acids assemble together to make a protein? Let's first take a look at the structure of each amino acid, okay? All 20 amino acids have the same backbone, which contains the amino group and the carboxy group. However, the R group shown here, also known as a side chain, differ in each amino acid. So the R group really determines the unique properties of each amino acid. Proteins are formed by linking amino acids one by one. Amino acids are linked through this called a peptide bond, which is a covalent bond formed between the two amino acids when the carboxy group of one molecule reacts with the amino group of the other molecule, releasing one of the water molecule. The peptide chain can be extended as amino acids being added up, and we always call the end with amino group as N terminus, and the end with carboxy group as C terminus. When we describe a protein sequence, we always follow the order from N terminus to C terminus, similar to the 5' to 3' in DNA and RNA molecules. Okay, now we learned all these components in translation. Let's see the major steps in translation. Same as transcription, translation also has 3 steps as initiation, elongation, and termination, all right? So during initiation, ribosome assembles around the target mRNA. The first tRNA, known as the initiator tRNA, attaches at the start codon AUG. And during elongation, the tRNA transfers an amino acid to the tRNA corresponding to the next codon. And then the ribosome then moves to the next mRNA codon to continue the process. And covalent peptide bonds are formed between adjacent amino acids, right? And then lastly, during the termination, when a stop codon is reached, the ribosome releases the polypeptide, mRNA and tRNA. So this is a picture of a translation process. We already talked all about it, so I'm not going to repeat. Okay, so during the translation, multiple ribosomes can make proteins on a single mRNA molecule simultaneously. So this structure is named as polysomes. So the advantage of polysome is increasing the efficiency of protein production. Shown in this picture is a typical polysome. So the strand in the center is a mRNA molecule, and many ribosomes are loaded on this mRNA. You can see the peptides made by all these molecule, this polysome, right? So based on this picture, I want to ask you a question. So can you tell where is the 5' of the mRNA? Is it on the top or on the bottom? Okay, so remember that the translation starts from 5' and moves to 3'. All right, so if the ribosome is close to 3', it would make a longer peptide chain. Showing in this picture, the peptide chain on the bottom, like here or here, is shorter than on the top. This is a very long chain. You can see this is longer, okay? So indicating that 5' is at the bottom and 3' is on the top. This is the mRNA, okay? Once proteins or polypeptides are made, they may not be functional because they may need further chemical modifications, or they may need further folding to become functional, right? Sometimes proteins need to be further processed to form a mature and functional protein. And during protein folding, amino acids interact with each other to produce a well-defined three-dimensional structure known as the native state. The correct three-dimensional structure is essential to function. Protein folding is often mediated by chaperone proteins. Chaperone proteins exist in all cellular compartments and interact with the polypeptide chain in order to allow the native three-dimensional conformation of the protein to form. Protein is a major component of our cells, and we make many types of proteins every day. For a collection of all proteins in a given cell type, we call it as a proteome. How big is the human proteome? Interesting, about 250,000 different proteins can be made from our 20,000 human genes, more than 10-fold more. We learned that alternative splicing can contribute to the numbers, and alternative transcription starting sites and the protein modifications can also contribute to the complexity of human proteome. Okay, so let's take a look at the protein modification. Protein modifications sometimes are called post-translational modifications, or PTMs, which refers to the covalent and generally enzymatic modifications of proteins following protein biosynthesis. Okay, so the PTM or post-translational modifications can occur on the amino acid side chains. Remember the side chain, okay, not backbone, or at the protein's C or N terminal. There are many kinds of PTMs. And this picture only shows the common PTMs. Most of these PTMs can only occur on specific residues. Residues mean specific amino acids, okay? For example, the phosphorylation showing here is a very common mechanism for regulating the activity of enzymes. And phosphorylation probably is the most common PTM, all right? Phosphorylation can occur on 3 residues, serine, threonine, and tyrosine. But then the ubiquitination showing here is another common PTM that regulates protein degradation and can only occur on lysine residue. Okay, you can imagine the combination of all these different PTMs on different proteins greatly increase the diversity of our proteome. So here I give you an example. We learned the cyclins and the CDK complex, right? So CDKs can only be activated when it binds to the cyclin. Why is that? Because the interaction of CDK and cyclin changes the conformation of CDK, and CDK becomes self-phosphorylated here. So this phosphorylation turns the CDK to become an active kinase. However, at a certain time in cell cycle, what happened is the cycling here is polyubiquitinated. This is called a polyubiquitinated chain, okay? So, and this polyubiquitination marks the cycling for degradation by the 20S proteasome, all right? So once cycling is destroyed by the proteasome, the CDK conformation changes back and become inactive again. From this example, you can tell that PTM, like a phosphorylation or a polyubiquitination, they are extremely important for controlling the protein activity or enzymatic activity. Okay, so the different levels of protein structure are known as primary, secondary, or tertiary, and the quaternary structure. The primary structure is the sequence of amino acids that make up a polypeptide chain. It always follows the N-terminus to C-terminus disorder, okay? Protein secondary structure refers to the regular repeated pattern of folding of the protein backbone. So the two most common folding patterns are the alpha helix and the beta sheet. We're going to later see a video. So here shows in this picture is a alpha helix. Then the tertiary structure refers to the overall folding of the entire polypeptide chain into a specific 3D shape, okay? Many proteins are formed from more than one polypeptide chain. And then that's the quaternary structure describe the way in which the different subunits are packed together to form the overall structure of the protein, okay? Here, this hemoglobin protein is shown. Let's watch a video on this.
Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body.
If genetic mutations causes changes on amino acid composition, some proteins can be misfolded and result in disease. Like shown here in figure A, this is a normal folding pattern of a prion protein. Most of the protein is in a helical configuration, alpha helices, right? And then shows in this red color. And then showing in B, this is a refolded protein. This is in a disease-causing configuration. In this form, most of the protein is in the beta sheets, showing this red color, the yellow color, sorry. This ribbon-like section. With a different structure, misfolded protein do not function properly and can result in an abnormal phenotype. All right. So in lecture 2, we introduced the protein called the FUS, right? Remember, the FUS protein has a mutation, and this mutated protein changes its structure and may contribute to the ALS under the stress. So, but here we're going to introduce another example, the cystic fibrosis. That's a great example to explain this protein folding problem. So we went through cystic fibrosis quickly when we talked about autosomal recessive diseases, but we did not really discuss what mutations can cause this disease, right? The CFTR gene encodes chloride membrane channel protein, and that is responsible for releasing the chloride ions out of the cells. The mutations of the CFTR gene affecting chloride ion channel function and leads to the dysfunction of epithelium fluid transport in the lung, pancreas, and some other organs as well. So the patient can have complications such as this thickened mucus. You can see those attached in the lung, and with frequent this respiratory infections. You can see this infection, bacterial infection, and also in the pancreatic inefficiency, giving rise to malnutrition and even diabetes. Let's see the CFTR gene mutation in the cystic fibrosis. In this figure A, you see this is the CFTR gene showing here. Okay, now you should be able to tell that this gene is how long? Okay, it's 250 kilobase pair long and has how many exons? Yeah, so from 1 to 24, it has 24 exons. Okay, when the gene is expressed as a full-length protein, showing in this figure B, this is a protein sequence. Okay, this is protein from, you can see this amino N terminal, and then carboxy is a C terminus. Okay, so this protein totally has 1,480 amino acids, sometimes just saying AA. Okay, and exon numbers, functional domains are all labeled here. So on the top, those are the exon numbers, and on the bottom showing here like the shaded different regions, those domains, you can say those are their functional domains. All right. The most common mutation here, this showing here is a delta F508. This is a result from a deletion. Delta usually means deletion, okay, of 3 nucleotides and lead to loss of this amino acid. F means phenylalanine at the residue, the position of 508. This is a 508 position on this protein. So this one single amino acid deletion produces a incompletely folded protease-sensitive form of CFTR protein. So protease, we know that the function of protease is chop the protein, digest the protein into pieces, right? So if it's very sensitive to protease, that means it can be rapidly degraded before even entering the Golgi complex for this protein. So that's the how this mutation will cause loss of the protein, loss of the function, and then that's end up you have this disease. Okay, that means protein folding is extremely important. All right, so that's all for today.